Okay, make sure I can be seen and heard, okay? Thank you. And oh my gosh, it's too many people are here. All right, I guess we'll start, even though it's a small class today. Yikes. Okay. Hopefully people will chime in. Yeah. Okay. So we're down to the last few weeks of the semester, of the short and condensed semester. So 3 full weeks, and we'll go to that last day, I'm sure. We'll go over the exams. I have not looked at any of the exams yet. We'll try to get those graded in the next 48 hours. Don't know what you all thought of the exam, but we'll see how you did on Wednesday. Another quiz this Friday. You're gonna have 24 hours to do the quiz. You have a final website comment next Wednesday, and then another quiz on the 13th. Lots of stuff. I'm really sorry about all that, but and then a final exam. 3 weeks, I guess. Yeah, 3 weeks, and you'll have a couple of days to do that. It will be due on the 20th. It's the final date is scheduled for the 25th, but you'll have it ahead of time because I don't want nobody wants to go beyond that Wednesday. So that's how we're going to do it. So it'll be due on the day of the scheduled finals when it will be due. Okay, and it will not be comprehensive, meaning it will just be over the last section. So from today, or from actually last week when we started talking about spoilage, to the last day of class. Okay. So we went through the principles of spoilage, and we're going through commodity by commodity, and we talked about fruits and vegetables. Conditions are good for all sorts of microbes, but because of the high pH, EH, and so forth, psychotrophs do particularly well. And I gave you that little general rule. When conditions are really good for everybody, bacteria win. They get a little bit less good, yeast win. And when things get really stringent, then if you're going to see spoilage, it will generally be by mold. So low water activity, low pH, so forth. When spoilage does occur for fruits and vegetables, they go by the name of, often go by the name of these rots, like soft rot. And soft rot is kind of classic, and the classic cause is a microbe called Erwinia kerativora. And other bacteria will cause very similar kinds of effects. So some of the other pseudomonads that we'll talk about in a second. It's common in all sorts of produce, and it's caused, the soft part comes from the degradation of pectin via pectinolytic enzymes. And pectin, as you know, is the structural carbohydrate that provides structure to fruits and vegetables. Okay, crispiness of vegetables and so forth is due to pectin. Okay, provides rigidity. That's a good way to put it. Once the pectin is degraded and the bacteria gain access to the inside of the tissues where the sugars and nutrients are. And that's why microbes produce pectinolytic enzymes to get to the good stuff, okay? To get to the good stuff. Okay, and it results in softening as well as the growth of those microbes on those substrates to produce acid flavors, odors, and defects. Okay, so Erwinia kerativora is a mesophile, even though it can grow at low temperature, so it's a psychotroph, but it's really a mesophile. And it's in the Anaerobacteriaceae, in contrast to the other rot or soft rot organisms, like the Pseudomonas, the Flavobacterium that we've talked about. Molds are notorious rot or blight producing organisms. And so blight is usually, I think that Dr. Helen Adams will refer to a blight. She's the mold expert. A blight is something that occurs in the field, whereas rot might be more what we might refer to something after it's been harvested. But there are a number of these, so some of these molds do both. They cause infestation in the field and they can cause spoilage in the food after harvest. So there's a bunch of these, and we'll go through these, and I guess you don't have to necessarily know them all by heart. The exam is going to be open book anyway, but these are the common ones. So botrytis is that typical one that you'll see on strawberries and grapes. Rhizopus is another one that you'll see on vegetables. Typically, I'll have a little bit more blacker than the botrytis, which is more gray. Then there's blue mold rots on fruits caused by penicillium. There's a watery rot on tomatoes. Generally, this mold kind of sticks to machinery real well. Alternaria rot. Again, there's a long list if you're into this sort of thing. I do want to kind of call out this one on potatoes. Phytophthora infestans causes a blight on potatoes, and you should probably know from your microbiology history what was the significance of that organism. Anybody know offhand? Phytophthora infestans? I want to volunteer why that one is a historical significance. Yeah, it caused the Irish potato famine. More than 150 years ago, in Ireland, they depended on potatoes as a staple, just like in other countries, they may depend on rice and other grains. They depended on potatoes. And this mold wiped out the potato crop over a period of years and led to mass starvation in Ireland. About 1 million people starved to death because of that. And another 1 million or so people left Ireland, came to the Americas, came mainly to the North America, United States. And that's why, and one reason why there are so many Irish Americans in the United States, because of that. Okay, and then lastly, just preservation. We've gone through all these things, so I'm not going to repeat this, but asepsis, washing, low temperature, and so forth. Okay, let's move on to another commodity, and that's meat products. Again, let's look at the composition. Meat, again, high protein, it's a great growth medium. Almost every microbiological media that we use in the laboratory has some sort of meat extract, beef extract, or something, because it's so good nutritionally for microbes. Fresh meat has high water activity, 0.99, a high EH, at least on the surface, and things that are mixed like ground beef have a high EH. And the pH is usually 5.6 in the meat. Now, pH of live tissue, muscle, is 6.5. You might know why this goes down. Why does it drop a whole pH unit from live to slaughter? If it's because, usually if I have an animal science student, they know the answer to this question right away. But in the live animal, as they metabolize sugars for glycolysis, they make lactic acid. So that's how your muscles work. They make lactic acid and make ATP, so muscles can contract. But in the live animal, the lactic acid gets into your circulation and moves away from the muscle. If it doesn't, you get a cramp. But in the animal that's been slaughtered, the enzymes are still active, even though the animal is not. There's no circulation. So the pH of the tissue drops a pH unit because of the buildup of lactic acid. Okay, and it does affect meat quality. 5.6 has a little bit, it's more inhibitory than 6.5, but not too much. Okay. We also know from our section on preservation that the keep out rule, which would be really nice to apply, but it's difficult because all the handling of fresh meat, if you've been in a slaughterhouse, and some of you probably have, one of the things that one of the discoveries that you'll make is, or one of the questions you'll think is, man, how do they make meat even as aseptic as it is? There's just so much stuff going on there that you wonder, you're surprised that there's not more microbes on meat. And it's getting better and better, actually. Okay, so meat is very perishable. And I did the state of the animal prior to slaughter, all the handling will influence the microbiota that's established. So the inner tissues, so the surfaces are going to be contaminated, but the inner tissues are essentially sterile. So the interior of a roast or a steak is sterile for the most part, but it's the surface and exposed areas that get contaminated. And they will get contaminated with a wide array of microbes. Some of those microbes are fecal from the evisceration process. Some of them are associated with the hide. Some are associated with the environment. And ultimately, which organisms will predominate and grow will depend on the temperature. Okay. So low temperature is going to select for cyclotropes, as we've talked about. But there's a lot of protein in meat, and so you might even get more proteolytic bacteria proliferate. Okay, what are the bacteria that are associated with meat? Again, it's a wide diversity. So lots of psychotrophic bacteria like the Pseudomonas, lactic acid bacteria, both psychotrophic and mesophilic, and then all sorts of other ones, spores, Clostridium, Bacillus, and then all these pathogens, coliforms and so forth, Salmonella, Staph, E. coli, a whole bunch of them. There are some psychotropes that we have not talked about previously that are specifically associated with meat, like Moraxella, Acinetobacter, Psychrobacter, Brachothrix. And these are psychotropes, and they can be problematic, partly because they grow cold and partly because they are very, they like growing on meat maybe more than other microbes. Okay. And then there's occasionally some molds and some yeast, and we'll get to the situations where they are important in just a minute. Okay. So which organisms predominate and the conditions of storage will dictate the type of spoilage that occurs. So I've been using this chart for a long time, and actually it works pretty well for providing you a general sense of which organisms will predominate. So meat is generally held under a few different conditions. So the meat that's in your refrigerator at home, if it's in just a tray with a plastic overwrap, will have a high EH because it's the plastic overwrap is usually oxygen permeable. The pH I just said is going to be 5.6-ish, which is conducive for microbes. The water activity is going to be 0.99, and if it's in your refrigerator, the temperature will be low. Okay. There's another condition where it's refrigerated, so temperature low, EH high, pH high, but the water activity is a little bit lower, and I'll explain the situation in a minute. But this is a situation where the meat is dried at the surface. Okay, it doesn't occur commonly, and I'll just give you an example in a minute. Another situation is refrigerated, temperature low, water activity high, pH high, but now it's anaerobic. This is the boxed beef that we talked about before, where the beef is processed in the slaughterhouse facility, put in cryovac bags, and then the air is flushed out, and then it's kept anaerobic. And this is that meat that has a really long shelf life, has the purple color. There's a really long shelf life, a month or more, okay? because of the low EH. And then there's a situation where it's also anaerobic, pH okay, water activity okay, but the temperature is high, mesophilic range. This would be like this meat over here that got temperature abused, and this is not a good situation because anaerobic mesophile spells Clostridium. Okay, so that's trouble. Okay, so let's now look at the examples of those situations. So here's situation one, aerobic, high water activity, pH 5.6, low temperature. These are the ground beef and the cuts in oxygen permeable packages that you see at the grocery store. It will favor those aerobic psychotrophs that we've talked about, Pseudomonas, Flavobacterium alkaligenes. Okay, you can get some of those other guys that are specific to meat, like Brachothrix. Okay, and what they will do is generally they're going to produce surface slime, pigments, lipolysis. This is your typical meat spoilage. You could tell by you putting your finger on that looks like a steak of some sort, a T-bone or something, and you'll feel slime, and it will be spoiled. You'll consider that spoiled. Usually it takes about 1 million to 10 million per centimeter squared. Okay, so 100,000, not so bad. Get to a 10 to the 5, you're starting to get borderline 10 to the 6. You'll probably be able to tell. Okay, and you may even smell it. Let's go to the second situation. Aerobic, lower water activity, and again, this is at the surface and low temperature. And this is really one scenario, and that's dry aged beef. Okay, dry aged beef. So any of you that have worked in high-end restaurants will know that when restaurants that I'm trying to think, maybe Misty's. Is Misty still open here in town? I don't even know what's open these days. But some of the high-end restaurants, Venue and JTK here in Lincoln, and some of the steakhouses that you know about that have chains like what's a couple of them I know of. Names, I forget the names now. Will feature dry aged beef, and what they do is they put, usually it'll be whole sides or quarters in a cooler, and with good air circulation, and the surface gets dry, and what happens is that meat gets tender and develops all sorts of nice flavors. But on the surface of that meat, you get mold. Remember, conditions good for everybody. You get bacteria. Conditions a little bit harsh, you get yeast. And when conditions get really severe, like dry on the surface, you get mold. And these are the mold: Cladosporium, Thamnidium, Sporotrichum. And what the restaurant will do, or whoever's aging that meat, they'll just trim that mold off. The mold actually can provide some tenderization as well, because the mold is producing the same kinds of enzymes that are produced in the muscle tissue that cause the meat to get tender. Okay, so that's the second condition. Third condition is the box beef, where I just said, vacuum packaged, long shelf life, but does favor psychotrophic lactic acid bacteria. So most lactics are mesophilic, but there are some that are psychotrophic, and those will be Lactobacillus sakei, other lactobacilli, Carnobacterium, Leuconostoc, and also that Brachothrix that I mentioned before, okay, which is a gram-positive psychotroph associated with meat, okay, and it's facultative, but likes those anaerobic conditions. Okay, so that's the third condition. And then the fourth condition is when you have a temperature abuse of those vacuum packed meat, and that will favor the mesophilic anaerobes. Sometimes you'll be lucky if it's lactobacillus, unlucky if it's Clostridium, because then you can get all sorts of problems, putrefaction and growth, and that could be trouble, especially if it were a CBOT. So that's the summary. These are the four conditions that we talked about, and these are the microbes that will proliferate or predominate: psychotrophic bacteria, psychotrophic mold, psychotrophic lactic acid bacteria, or mesophilic Clostridia or lactic acid bacteria. Okay. Processed meats, we'll just spend a minute or two here. These are generally much better preserved, fewer spoilage organisms because we have nitrite to inhibit Clostridia. We have salt to inhibit gram negatives. Water activity and pH can be lower. Other antimicrobials could be present. Nonetheless, for processed meats, we're usually talking about cured meats. And so one of the spoilage problems could be the degradation of that nice pink cured meat color that we like. And that can be degraded by peroxides. Some lactobacilli will produce H2O2, and that will cause that nice pink color to become oxidized and look green. And you'll see, sometimes I even get calls from friends and consumers. They'll say the meat tastes fine, it doesn't smell, but there's like a green sheen to it. What's that all about? And it's because some of the lactobacilli produce peroxides, and you get this green, kind of almost fluorescent-like pigment that's formed. Okay, if it gets, if they start really growing, then they can start producing some souring. Usually, you don't see that. Okay, and again, this just shows that nice pink color degraded to form green or other kinds of unpleasant, undesirable colors. Okay. Preservation. Again, we've talked about the all those keep out things in meat, asepsis, carcass washing, low temperature, modified atmosphere, and so forth. Okay, now we move on to, so we've covered cereal and grains, produce, meat, and now we're on fresh and processed egg. And I have to tell you that for eggs, every year I think, egg spoilage is so rare. Now that's separate from egg contamination with salmonella, which is not rare. But spoilage in and of itself doesn't occur very often, and it's because of the advances in the egg on the production side and on the egg processing side. It's just not very common yet. I keep it in here because it's for reasons that we'll go into in just a second here. Okay, so composition, again, favors spoilage. The water activity is high, eggs are nutrient rich. But as you know, eggs have lots of barriers. Okay, so obviously the structure of the shell, the cuticle and the inner membrane is a major barrier. The pH is a barrier, but in this case, most of the foods that we've been talking about have a pH below 7, and you get into the acidic range. That's a really formidable barrier, but eggs have a higher pH, and high pH can be a barrier. 7.5, not so much, but 8.5, most microbes do not like pH 8.5. And the white of an egg can get to 8.5 after storage. Eggs have lysozyme, and we talked about lysozyme earlier when we talked about some of the natural antimicrobials. Lysozyme is an enzyme found in egg that degrades the cell walls of microbes, and it's naturally present in egg white. And in fact, it's one of the main sources of commercial lysozyme is to extract it from egg white. We use it, again, in the laboratory all the time to lyse cells, to collect DNA. And so eggs are a great source of lysozyme. Egg whites also have these three proteins, canalbumin, avidin, and apoprotein. And these are, they bind vitamins. So avidin binds biotin, apoprotein binds, I think it's riboflavin, canalbumin binds, not a vitamin, binds iron. And so by binding those nutrients, it keeps them away from the microbes that want those nutrients. Okay. And so, well, a good question to ask. Why does the hen put all of these structural and chemical barriers in the egg? obviously the egg is intended to be a chick. Okay, and so the hen, nature, puts all of those barriers in there to protect the chick from infection. Okay, in our case, it's the edible egg that we're interested in, but that's why these barriers exist. Okay. Okay, so despite the barriers, if you've been to a poultry farm where they're producing eggs, there's a lot of microbes around because there's lots of bird poop in these environments, and so there's lots of exposure. Okay, so that's one factor that accounts for spoilage. The other one is that shell, the cuticle, the inner membrane, are porous. They look like they're not, but they are, and bacteria can penetrate into the interior. Again, once they get into the interior, they still have to deal with lysozyme and all those other things. Okay, but in an egg producing environment, if the humidity is high and the shell is moist, you can get penetration. Okay, so here's the anatomy of an egg. So here's the yolk, here's the shell, and you can see all these layers around the shell. There's an inner shell membrane, outer shell membrane, and so lots of barriers inside here. If we blow the shell up, cuticle, shell, egg white on the inside. So there's a membrane here, cuticle, but here's a pore. It's small, but there is a pore. There are pores in that shell. And theoretically, microbes, if it's humid enough, can transverse this. Okay, so the surface of that egg may contain very low, from 100 to 100 million, lots of different genera represented. Again, cycrotrophs for sure, because we've selected for them at low temperature. Coliforms for sure. One that you probably haven't talked about much is called Proteus, is kind of, that's associated with eggs, just like that Brachythrix is associated with meat. And then you can get pathogens like Salmonella and Listeria. And you can get also Bacillus, especially in liquid egg products, which we'll get to in a minute. And you can also get yeast and mold. Okay. Again, we come back to that rot situation where we call these rots. And that's just this little caveat here. 50 billion shell eggs consumed each year. That's a lot of eggs. You could do the math in a population of 350 million. That's a lot of eggs per person. But spoilage is uncommon. Okay, if it does occur, there are several microbial rots: green rot, colorless rot, black rot, fungal rot, caused by the characters that we just talked about, Pseudomonas, Proteus, and some of the molds. I don't know if Dr. Auchton talked about trans-ovarian contamination with Salmonella. So if the bird is infected with Salmonella, it's possible that Salmonella could be implanted inside the egg as the egg is being built, if you will, through trans-ovarian contamination. And this is thought to be one reason why there's so many egg recalls, is because of trans-ovarian contamination, but can also occur by the conventional means that I talked about. And there are lots of egg recalls every year, and there usually are large recalls because by the time it's discovered, some of these egg farms produce millions of eggs in a short amount of time, and so the recalls can be pretty large. Now, I did mention Bacillus. I said I was going to come back to it, so I wanted to mention something about Bacillus and pasteurized egg spoilage. We'll come back to the Salmonella in a second. So a lot of the eggs, you might be surprised to learn, so there's shell eggs and then there's processed eggs. So shell eggs are exactly what they sound like, eggs that you buy out of a carton. You take them home, restaurants will use them, and you crack them open and you use those eggs. In the food industry, things like cake mixes and pancake batters and frozen waffles and all those kinds of things that use eggs as ingredients, they do not buy shell eggs and crack them and process them. They buy liquid egg, okay? And that liquid egg is basically whole egg that goes to a processor. They crack the eggs and they pasteurize them. So they basically scramble those eggs up and they pass them through a pasteurizer at temperature and times that we talked about before that satisfy the FDA, but that don't cook the egg. So you can't pass that egg through a pasteurizer and get cooked scrambled egg out of there. That doesn't do anybody any good. But if the egg is flowing and the time is short and the temperature is not too high, you get that liquid egg. And it could be whole egg, it could be egg white, it could be egg yolk, pasteurized. Okay, and that's how that's done. But there are, just like we talked about for milk, there could be some heat-resistant organisms. In this case, they're heat-resistant bacillus that can be a problem in those liquid eggs, and that does come up. So that's a spoilage situation. It's not the shell egg, it's the liquid egg, and that's not uncommon. Okay, I did want to say something about the salmonella. So again, 100 billion eggs per year. Maybe 1 in 10,000 are positive for salmonella, but if you do the math, that's 100,000 eggs may contain salmonella. Okay, so this is a problem. And so the FDA has estimated 80,000 cases of salmonella food poisoning due to eggs. You see these big recalls. Let's see this one here, 200 million eggs recalled. Okay, and they're often, there's another one, 207 million eggs. And they usually are large recalls involving many states. So that's why it's important to have this section on egg spoilage, not so much for the shell eggs, but for liquid eggs that spoil by a bacillus, and for the salmonella contaminated eggs that cause these big recalls. In fact, I think it was, yeah, this one was a couple of years ago, but I think it went all the way into Thanksgiving. And so that was really concerning because a lot of people use eggs for different things around this time of year. Okay, and then we talked about preservation keep out all those rules. One of the reasons why shell eggs do so well these days is because they usually have a chlorine treatment. They're stored at the right temperature. You could even pasteurize, I should have mentioned this, you can pasteurize eggs in the shell. So there's a couple of brands that do that, and they pass them through hot water, and you'll see those eggs. They cost a little bit more, but for chefs that want to use raw egg, like for hollandaise sauce and salad dressings and things like that, you should buy those shell, or eggnog this time of year. People like eggnog. If you buy the pasteurized in the shell, then you could use those eggs. Otherwise, you should not use raw eggs. And then the other way, of course, to preserve them is through pasteurization. There are some salting and techniques that can be used. You could freeze, but usually they'll often still be pasteurized and then frozen, pasteurized and then dried. Okay, so now we move on, staying kind of in the poultry family, talking about poultry, which is very similar to meat. So we won't spend too much time here, but the difference with poultry that's different from meat is the greater opportunity for cross-contamination. Now meat, so beef and pork and lamb and others do have lots of opportunity for cross-contamination because the carcass in the slaughterhouse passes on conveyors and then goes on and then gets cut up and so forth. But in poultry, there's even greater opportunities for cross-contamination due to how birds are slaughtered. Okay, so they will go through, for example, defeathering operations. They'll go through chill baths, all sorts of opportunities for cross-contamination because the birds are passing through common equipment. So even if only 1 bird in 10 tests positive for salmonella, the next bird that goes through the defeathering step will become contaminated. Same thing in a chill bath. So the birds are slaughtered and they want to get the temperature down as quickly as they can. So they might throw all those birds into a chill bath and now they're all cross-contaminated. Of course, the other option is not to use chill bath, but to use air chilling, and that's become very common. That's how they do it mostly in Europe And that's the way that Smart Chicken, you may have seen Smart Chicken, says air chilling. Okay. The microbes that we worry about in poultry, again, are the same spoilage organisms, cycrotrophs, lactic acid bacteria, but also pathogens. And of course, Salmonella and Campylobacter are the ones of greatest concern. And the other thing that's different about poultry is most poultry is processed with the skin still on the bird, and bacteria have evolved and done a really good job of sticking to the poultry surface, and it becomes hard to dislodge them. Okay. So again, pathogens, spoilage organisms, and we notice the defects similar to how we do for beef and pork: slime, off odors, and maybe formation of pigments. And then there's the preservation strategies, again, similar to meat. So keep out, remove, all those things. And in poultry, the keep out rule even applies to on the farm. So what's becoming very common these days is to try to use probiotics and prebiotics, which we'll talk about in the next unit, to keep to competitive, to outcompete pathogens in the GI tract of those birds. And so that's been done, and with some success, to try to keep the pathogens out of the flock. In Europe, not so much in the US, they will even cull, that means remove infected birds. So you could have a whole flock at a farm that's Salmonella free or Campylobacter free. Okay. Not so much in the US, to my knowledge. Okay, so now we leave eggs and poultry and move on to fish and seafood. Again, composition strongly favors spoilage. So we have high water activity, neutral pH, very aerobic, especially if it's fish that's been filleted and so forth, very aerobic, protein rich. Some fish has high in lipids, and some seafood is high in carbohydrates, especially mollusks. And then, as I just mentioned, processing like filleting increases surface area, but also exposure to evisceration. When you pull the guts out of a fish, you're getting exposure, deboning, filleting, all increase surface area. And there's cross-contamination, knives, water, ice, equipment, direct contact. Fresh fish, if any of you do some fishing, and I know many of you do, here in Nebraska, it's a very popular recreation and sport. Fresh fish, and if you don't fish, but you like fish and you go to the fish market, you're looking for eyes clear, bright, and full, gills bright, flesh firm and moist, good fish odor, in contrast to spoiled fish where the eyes are kind of dull and shrunken, cloudy, and the flesh is pale and soft, and it may even be slimy and fruity. That fish you don't want. Okay. And spoilage is related to storage time. So if kept cold, you maybe have, maybe a week for fresh fish, depends on how, if it's been filleted and so forth, often less than that. So I like fish. And when I go to the grocery store, I'd actually almost rather know that the fish had been frozen and maybe put in the fresh case the day before, rather than fresh from the get-go, because it's Nebraska. If it was caught fresh in the Pacific or Atlantic, by the time, especially if we're talking about marine fish, by the time it gets to Nebraska and gets to the local Hy-Vee, boy, you're lucky to get here in 7 days or within 7 days. So a lot of grocery stores, even if they say it's fresh, it's arrived frozen, and then they keep it frozen and they put it out in the fresh case. And the reason for the short shelf life is partly microbial, but it's partly because of enzymes in the tissue that degrade the proteins through protein autolysis by the enzymes in the tissue. So in meat, so if we jump back to where we were talking about tenderization of meat, and why aged meat is more tender is because there's proteolytic enzymes in the muscle tissue that degrade protein, muscle protein, and make meat tender, which is a good thing. In fish, we don't want tenderization. We want that fish to be, the protein to be firm. And so those enzymes, those endogenous enzymes will make the protein, will hydrolyze the protein, and then those amino acids can be metabolized by bacteria. So even if the bacteria weren't there, the endogenous enzymes would cause texture problems, but the amino acids then get metabolized by bacteria and produce all sorts of things, trimethylamine, ammonia, H2S, and these things called biogenic amines, which we'll talk about in a minute. So it's the combination of endogenous enzymes and bacteria that degrade those amino acids. Okay, so again, there's selection for psychotrophs. Fish generally are growing in cold water, so there's psychotrophic bacteria affiliated with the fish. Fish from temperate climates might have some more mesophiles, so maybe fish from the Gulf of Mexico, for example, or from tropical areas might have more mesophiles. And then you have shellfish, which have a specific group of microbes because some of these shellfish are filter feeders. They filter water, collect the food. So then you could get viruses. Again, some of the fish from the Gulf of Mexico, sometimes a lot of those shellfish can have mesophilic organisms that could include viruses. And some of the fish like lobster and so forth have lots of appendages and they could trap microbes as well. And then how fish is caught, line versus nets, that may have an effect as well. Okay, so I mentioned the difference between the looks of fresh fish and spoiled fish, shelf life 7 days, and what happens in addition to the protein hydrolysis is you can also have lipid oxidation occur, and that's more of a chemical or biochemical function. These are the two main phenomena: protein hydrolysis and amino acid metabolism. Okay, I mentioned biogenic amines. Biogenic amines are formed from a few of the amino acids that are found in fish protein: histidine, tyrosine, there's a few other ones. And the microbes that produce decarboxylase enzymes, histidine decarboxylase, tyrosine decarboxylase, take the free amino acid and make an amine, histamine, tyramine. And these are called biogenic amines because they are biologically active and can cause a form of food poisoning. It's not severe, nausea, headaches, sometimes other complications, but these are amines. And so especially some people that are sensitive to these histamine and tyramine will suffer the consequences. Okay, organisms that produce these decarboxylases include lactic acid bacteria, and a microbe that's particularly associated with this group called Morganella. So we've learned 3 new groups of organisms, or 4. We learned Erwinia, that's associated with soft rotted vegetables. We learned Brachothrix, which was associated with spoilage of anaerobic meat. We learned Proteus, which was associated with eggs. And here's another one, Morganella, associated with biogenic amine formation. Now, when we talk about fermented foods have a lot of these biogenic amines as well, because fermented foods, especially like aged cheese and aged dry fermented sausage, have a lot of the proteins break down. They have the same kind of etiology. Proteins break down, amino acids form, amino acids get metabolized. So sometimes you'll hear people complain about the same kind of symptoms in aged cheese and dry fermented sausage and others. So we'll come back to that because lactic acid bacteria can produce these. Okay, other spoilage effect of eggs, trimethylamine, TMA, sometimes considered characteristic for fish spoilage. Again, you see these ammonia, putrefactive end products. That's anaerobic spoilage. Remember, we talked about that. And then rancid fruity flavors, and then sometimes sourness, depends if there's carbohydrate there. And then again, we talked about preservation. We talked about washing way back when, but washing, especially with maybe slightly chlorinated or acidified water, can help. Okay, and I think we're getting to about the second to last category are dairy products. Again, composition of milk is great for microbes, and so often we will grow microbes in milk when we want to collect and propagate them. High water activity, neutral pH about 6.5, EH is high, it's protein rich, carbohydrate rich, lipid rich. It's got everything that microbes like. There are some natural barriers. Lactoperoxidase is present in raw milk. There's some lysozyme, there's some lactoferrin binds iron, agglutinins binds cells. Unfortunately, when we pasteurize milk, these are all inactivated. So raw milk is susceptible, but also pasteurized milk. Okay, and we talked about raw milk contamination with pathogens when we talked about pasteurization. Okay, and we're going to talk about raw milk in a moment. And this is now a public health issue because a lot of people like raw milk, even though we talked about most milk in the U.S. is pasteurized. There are still some raw milk consumers. So what's in raw milk? So milk, whether it comes from a cow or from a human, is sterile when it's produced, but as soon as it leaves the cow, it's immediately contaminated by the microbes on the surface of the udder and in the equipment and so forth. It quickly becomes contaminated. Low temperature, and in most modern dairies, the milk gets cooled quickly. So, but you're still selecting for psychotrophs, and all sorts of other whole array of microbes that live in the animal environment, coliforms, LABs, and these pathogens. All sorts, just about every pathogen imaginable can be in raw milk. Okay, and that's why raw milk is almost always pasteurized, especially if it's going to be used for fluid purposes. The exceptions would be certain cheeses. And that's why we have the pasteurized milk ordinance, which tells us not only the conditions for how that milk should be held, conditions for pasteurization, but it even establishes limits. Okay, and it's very few foods in the United States have microbiological limits. Milk is one of them. Okay, and so the limits for raw product. Okay, so there's no limit for how many microbes can be in raw broccoli, raw anything, but there is for milk. And so if that milk is to be used for fluid purposes, i.e. drinking purposes, at the farm, it has to contain less than 100,000 CFUs from an individual farm. Now, once that milk is from a farm is mixed with milk from another farm and gets to the dairy in a big silo, they allow a larger amount because they realize that the pooling and so forth could increase that number. Okay, and we'll come back to those numbers in a few minutes. So raw milk spoilage by psychotrophs, including Pseudomonas, psychotrophic LABs. If it's pasteurized, then you can get thermoduric microbes. So we talked about bacillus, maybe even penibacillus can also occur. But most pasteurized milk is spoiled via post-pasteurization contamination. So if you see psychotrophs in pasteurized milk, it wasn't because the psychotrophs survived, it's because it became contaminated afterwards. Okay. Now, here's some more numbers. 100,000 or 300,000 in the raw milk. In the milk that's pasteurized, less than 20,000. So this should say pasteurized less than 20,000 in the pasteurized milk by the aerobic play count. coliform less than 10. And because usually the dilution is a 10-to-1 dilution, it really means none detected, okay? We say less than 10 because that's usually the first dilution plate, okay? But technically, you should not have coliforms in raw milk because that could signify, a coliform is an indicator. It could mean that there's E. coli or salmonella. So less than 10 basically means none. Okay, so spoilage can occur from, sorry, talking about spoilage of raw milk and pasteurized milk here, and spoilage of dairy products like cheese and butter and so forth. Usually it's caused by using poor quality milk, but there's also opportunities for contamination of those products, especially, cheese could have mold, yeast can be in yogurt and other products, but for the most part, the product will reflect how the milk was held. Okay. And the spoilage of milk usually results in fruity, bitter, rancid. If it's lactic acid bacteria, remember we talked about milk that spoils by lactic will be sour, milk spoiled by saccharose will be fruity or bitter. Occasionally you could have gassiness from coliforms. Okay, and then we talked about all the pasteurization things for milk, low temperature pasteurization. We'll get to fermentation next week. Okay, so I want to stop here, but I just wanted to note that what we'll talk about on Wednesday, I want to have a few minutes to talk about raw milk for public consumption, and this issue about which comes and goes, but it remains a popular topic for consumers. They want their raw milk, and we'll talk about why that might not be such a good thing. Okay, so we'll stop there, and we'll spend the first few minutes on Wednesday reviewing the exam. Okay, which I hope you've all done well on. Okay, thank you. See you all on Wednesday. Hopefully we'll all be here following the election on Wednesday.